Hello, this is Mr. Young of Anatomy and Physiology. Welcome to our discussion of cells and tissues. We are going to be focusing on, in this unit, histology, or the study of tissues. And in a period of 2 weeks, we'll try to hit on aspects that you will otherwise be spending entire semesters on in your histology courses in college. But as we begin today, we are reminding ourselves that we've been focusing on how cells get an idea of what they're going to become later in life. From the undifferentiated stem cells, whether it's toti or pluripotent, that are characteristic of the early embryo, to specialized cells that form the 210 plus different cell types that we find in the body. For now, we're going to be focusing on 4 specific tissue types of the body. And as we consider tissues, we've noted that cells can develop specific structures and functions that, when similar, can congregate to form a tissue, which is the next kind of order of progression in the organization of life, cells giving rise to tissues. So the 4 primary tissue types that we will focus on in lab combined include epithelial, connective, nervous, and muscle tissue. In our time today, we're going to be focusing strictly on epithelial tissue, its forms, its functions, examples of where it's found in the body. So to begin with epithelial tissue, I want you to consider the root epi as meaning above or upon. That's right, your roots are paying off here. Above or upon what? Well, think about the areas where we're going to find this. Above or upon the body, so you might think your skin as a covering. Linings of the body, so internal coverings, epithelial tissue. And lastly, glandular tissues. And so far you might be linking up the word gland with secretions. Well, secretions as well as protection, absorption, filtration, those are all key functions of epithelial tissue in general. So now let's try to link up our functions with the actual structures of epithelial tissue. And as we do that, let's start by considering key characteristics that distinguish epithelial tissue from the other tissue types, connective, muscle, and nervous. Let's begin with this. As you look at epithelial tissue, one of the things that should stand out is the fact that cells fit together like a puzzle. They abut one another. So that is distinct because when you consider connective tissue, for example, cells are isolated and embedded in an extracellular matrix. So when we talk about them fitting together, we can consider a cell right next to another cell, right next to another cell. They're linked together. Sometimes they're linked through junctions that connect them, specific molecules serving to connect them. Sometimes they have gaps that allow for communication between the cells, but regardless, they fit closely together. Second key characteristic is that because these cells are lining and covering parts of the body, that means that they have a surface that's exposed, that we refer to as the apical surface, or the apex, and a surface that is attached. That attached surface we'll call the basement, or the basal. Right? Basement membrane, basal surface, is that area where our epithelial tissue connects to the rest of the body, which typically will be connective tissue. Another unique feature about this tissue is that there's no blood supply going to it. Problem, you might think. How can it get, if it has no blood going to it, the nutrients that it needs in order to survive, in order to operate optimally? Well, we'll find that there are some unique adaptations these cells have to allow for them to get rid of waste products to products that they need, including particularly the process of diffusion. But the root avascular, or the term avascular, simply means without, as you've looked at your roots, specifically blood vessels, no blood vessels. Lastly, if we are having tissue that is exposed to outer surfaces, whether it's inside the mouth or on the skin itself, these have the potential to be damaged by abrasion, by different chemicals in your foods that you eat. And so we need to have the ability to regenerate, which means to replace that tissue with the exact same tissue. You might think about at this time the role of, that's right, stem cells in doing just that. The ability of an undifferentiated cell to differentiate to become a needed cell type. And it turns out we have some of those stem cells that are able to do just that. To begin with, let's address some vocabulary. The key vocabulary I want to hit on here involves the numbers of layers of cells in epithelial tissue. In the first diagram, you see a single cell layer, one cell deep. We have an apical surface above. This is the exposed surface. And we have a basement membrane down below, shown here, the attached surface, connective tissue down below. Whenever we talk simple epithelial tissue, we're referring to simply one layer of cells. Let's contrast that with the bottom diagram, which shows multiple cell layers. Still epithelial tissue, it's lining and covering the body. Again, basement membrane down below. This is where the epithelial tissue attaches to the rest of the body, connective tissue below. Apical surface above, exposed, attached surface. We call it the layered tissue, stratified tissue. If you're to study the stratigraphy of rocks, for example, you're studying layering of rocks. Here you're studying the stratigraphy of tissue. So stratified, more than one layer. When we identify epithelial tissues, then we will first identify whether it's simple or stratified, but then we'll have to consider the shape of the cells themselves. There are three specific shapes, beginning with this flattened or squamous cell. Squamous cells, as you can see here, appear to be smashed. For cuboidal cells, the name says it all. These are cube-like cells. They are shaped like a box, although they can be difficult at times to identify on slides. I want you to note very carefully the location of the nucleus relative to the outer parameters of our cell. It is right smack dab in the middle. And when you see these in the context of other cuboidal cells, those nuclei will be rather equidistant from one another and right in the middle each time. Also, take another look at just a perspective. It looks like a hexagonal cell above, and yet from the side it looks like a cube shape. Finally, we're identifying a rectangular cell shape on the bottom here. We refer to it as a columnar cell. And when you're ever wondering, is this columnar or cuboidal, if there's ever any doubt, one big clue is the location of the nucleus. Notice how the nucleus favors one side of the cell. It's not right in the middle. Turns out it's going to be favoring the basal surface of the cell. Our basement membrane will be down below. Apical surface up above. This is the exposed surface. So those are our three different shapes of cells that we'll typically be using. And when we, again, identify tissues, we'll have to combine both the shape as well as the layering, whether it's simple or stratified. So let's start by identifying some tissues in the body, their associated organs, their specific functions, and how the function relates to the structure. So if you identify this organ, you'll see that this is your lung, your left lung, your right lung, and several different lobes of it. And if you were to be asked about the function, you might describe the role of gas exchange. Remember, we are having to take in through the lungs helpful gases like oxygen and getting rid of waste gases from cell respiration, CO2. In order for gas exchange to occur at an optimal rate quickly, what kind of layering do you think we need? First, simple or stratified? If you said simple, you're exactly right. We want simple layering. And finally, look at those three cell shapes we had, squamous, cuboidal, and columnar. Which one provides for the most shallow flow of gases in and out? You got it, simple squamous. This diagram shows us below. Simple squamous epithelial tissue. This is looking from the top down. Here's a little nucleus of a squamous cell. Notice it's very flat. We are looking at the apical surface right now, up above. Down below, we see the basal surface, basement membrane. This is where the epithelial tissue is attached. So simply a single cell layer of flattened cells. Again, for specific functions, we are looking for exchanging of gases in the case of the lungs. So you can be thinking of absorption as well as secretion. Finally, note some other locations. If you stick to the concept of gas exchange, the lungs pick up oxygen and deliver it where? To body tissues that need it. How? Through carrying oxygen into the bloodstream and delivering it in the capillary beds. Capillary beds are simply those areas where the arteries then begin to narrow and stenose to a much finer blood vessel called the capillary, through which gases will eventually be exchanged and then delivered back to the venous system And that systemic circuit will carry through the venous supply blood back to the heart, to the lungs, and kick it out through simple squamous epithelial tissue. Let's look at our next diagram of tissue. Here we see epithelial tissue again, lining and covering. I'm looking at the apical surface above now, basement membrane, basal surface down below. Again, this is made by both epithelial cells above as well as connective tissue below. How many layers do we have? That's our first question. That's right, one. We're looking at simple epithelial tissue. Next, we consider the shape. What shape are we looking at? Notice the nucleus right in the middle of the cell. This is cuboidal epithelial tissue. So here we see it in the location of the ovaries of the female reproductive tract, covering the ovaries. Again, in the ovaries, we are secreting eggs, so you might think of secretion as one major role here. But in addition to the ovaries, we can find this in places like kidney tubules. And if you consider the role of the kidneys in the urinary system, you'll find that they have specialized tubules, shown here if I outline this one, which have the role in filtration. And as liquids pass through this canal, shown right here, certain things will be absorbed into these cells, and other things will pass right through. But if you look at all the cells that line this tubule, you'll see distinct kind of shapes here of a cube. Nucleus right in the middle. This is cuboidal tissue, simple cuboidal. So in addition to kidney tubules and ovaries, also specific glands and their ducts, secretion being a common theme among their functions. Let's contrast those last few with this cell type. Again, we are looking at epithelial tissue. It's lining and covering, and this time the digestive tract. Here's our stomach, which is now dumping into the small intestine. And the small intestine will navigate itself around several meters until it dumps into a sphincter that leads to the ascending colon, out through the transverse colon, down through the descending colon, and out through the rectum. So you consider what in the heck is going on here. First, we're certainly wanting to absorb nutrients, so absorption is going to be a key role, absolutely. But we also want to lubricate the wastes that are dumping out the colon. Lubrication, secretion, you might be thinking of. So we're looking at a simple epithelial structure. The shape is giving away our term of columnar tissue. And that columnar tissue has a basement membrane below. This is the basal surface, connective tissue below. And the apical surface, shown right here. So to just remind ourselves, tall cells, they've got embedded within them typically cells that have a bulbous or balloon-like look. And they will have within them mucus. And this mucus they can eject out to lubricate the lumen, or the center, the inside of your gut, so to speak, to help your fecal matter continue on its way. Let's contrast those cells now with this type of epithelial tissue. First, again, we'll consider our different layers. It appears right now, if you only look at the nuclei, that we have a band of nuclei right here and a band of nuclei below, which might suggest to you we're looking at stratified epithelial tissue. However, if you look carefully at any one of these cells, you will find that the cell extends from the apical surface above all the way to the basement membrane below, a single cell. So it looks stratified, but in reality it's not. We call it pseudo-stratified, or if you recall your roots, we are now looking at false layering. It's not really layered, it just appears to be. Where are we going to find this stuff? Well, if you recognize this diagram, you are beginning to look at the outer parameters of a lung. This will be your left lung. And we'll find that as you intake air and it flows through, this being your voice box, your larynx, it descends through your trachea, this rather hard kind of cartilaginous ring structure that provides a structure for your airway. This dumps into primary bronchi, then the bronchioles, and eventually alveoli, where you have simple squamous epithelial tissue. But in this area, we have incoming, in addition to the air we breathe, particulate matter. And that particulate matter will need to be sifted through and sieved through. Well, along comes our pseudostratified epithelial tissue. It has specialized structures on the surface of the epithelium, specifically the apical surface, that are seen here. These are cilia. Cilia are hair-like extensions that, in our case, are able to capture those particulates and are able to move them in a direction upward against where the air is actually flowing. We don't want that fluid. We don't want the particulates in the lungs. Problem. And so as we push them upward through the cilia moving like fingers, then we produce this phlegm that you're familiar with. This diagram also shows a great picture of our goblet cells. Again, goblet cells containing mucin, this enzyme that also has a watery substance with it. And the goblet cells exocytose their products into the respiratory tract to help eliminate and to help make that particulate matter able to be moved upward. So secretion, major role. In addition to absorption, we're absorbing different amounts of water to maintain water balance in this area. So let's stop and take a quick review of different epithelial tissues that we've already seen, simple epithelial tissues. We have noted that here in the trachea, if you look very carefully at the trachea shown in this diagram, we are identifying how air can pass through the larynx, the voice box, go down into the lungs, and here we have pseudostratified epithelial tissue that is on both sides here that serves to move our particulate matter up. We've also seen the presence of linings inside of the mouth. We have linings inside of the nasal passages. So we must have epithelial tissue. Here we have linings on the outer surface of the body, the cutaneous surface or the skin. In each of these areas where you have linings or coverings, we're talking about epithelial tissue. But whereas we focused on the simple epithelial tissue, for example, in the alveoli of the lungs, which was needed for gas exchange, and the simple pseudostratified columnar of the trachea, do you think we're going to encounter simple epithelial tissue when it comes to the oral pharynx? Think about all of the exposure that you have to acids, to chemicals, different enzymes, both from your own saliva that you spit out, as well as from the foods that you eat. If we had a simple layer of tissue, we might have problems with having to continually replace it time and again. As it is, the cells inside of this area, inside your cheeks, for example, replace themselves every 3 days. But we need to have more protection. Now we look at stratified epithelial tissue. Take a look at what will become your oral cavity here down through your esophagus below, and you see a layering of different epithelial tissue. Still lining, in this case, your oral pharynx, but now it's multiple layers of epithelium connected to a basement membrane below. Again, identify apical surface. You should be able to do that. Identify basal surface down below. And then consider, well, what shapes do we have? That doesn't fit cuboidal, perhaps down below. Overall, it looks more flattened. That's right. We're looking at squamous epithelial tissue. Again, stratified for the use of protection in the body, and so found in areas where it's exposed to a lot of friction. Here you see in the micrograph all of those squamous cells from here to the basement membrane below, connective tissue beneath that is also helping to form the basement membrane. So this would be the outer surface then. In fact, this would be a great example of not only your esophagus, but also parts of your skin, including the palms of your hand, which has multiple layers, as well as the soles of your feet. We'll talk later about the epidermis and how there might be some extra layers within that epidermis to define whether it's from those parts of your skin or from your arm, for example. Let's consider this picture now, which shows an obvious defined border, so suggesting lining and coverings. This would be the apical surface of these cells. Basal surface is now here, connective tissue here. But now look, we have several layers. If you look at the cell shape, take for example this cell right here. That's right, we're looking at cuboidal, but it's not a single layer, it's several. we're looking at stratified cuboidal. When it comes to the stratified epithelium, stratified cuboidal as well as stratified columnar will be relatively rare in the body. Where do we find it? In areas where we still have the role of secreting products like sweat and mammary glands. Mammary glands specifically secretions of milk. And now we have our final stratified type of epithelial tissue. Here we see our columnar cells, and we'll note the location of the nucleus at the basal surface. This would be the apical surface then, basal surface beneath, basement membrane below. So this is stratified columnar. Where do we find this? Again, still in locations where you're needing secretion as well as absorption, pharynx, salivary glands, anus, the urethra that extends out from the ureters to deliver the waste products of the urinary system. One final tissue that we need to address is a bit of a catch-all. Why a catch-all? Well, if you had to identify the specific cell shape here, you might say that's somewhat columnar, although the nuclei in the middle looks more cuboidal. Certainly more cuboidal here, and at the top it almost looks somewhat squamous-like. Well, in areas that are subject to a lot of stretching, like the bladder, think about the change in volume that you experience in your bladder. We need to have a tissue that can withstand that stretching, and we do, as it turns out, and we refer to it as transitional epithelium. Transitional because this tissue has to undergo the stretching, and at different times it'll look like even simple epithelial tissue when that bladder is extremely full and it stretches out. When compared to when there's little volume, you can clearly see all of these different cell layers within. So it is multi-layered. It is a stratified epithelial tissue. We refer to it as transitional, though, because of how it changes its specific appearance based on whether it's full or not, the bladder we're speaking of. So we end with our final type of epithelial tissue, and that is glandular epithelium. When you look at the term gland, it's referring to a secretory organ, something that secretes a specified product. Well, that term secretion is linked to a special root, that root crine. So now you wonder, all right, what system usually refers to in the body the secretion of products and has the term crine? That's right, the endocrine system. Well, glands likewise have this term crine, but we can differentiate them into endocrine glands that specifically secrete inside of something. I want you to be thinking of secretions inside the body, specifically hormones. So hormones produced perhaps by glands and then having to be secreted into the bloodstream, into interstitial fluid and into bloodstream that get delivered to specified target areas. But they're ductless in terms of not having ducts that extend them to the outside of the body. Contrast the endocrine now with the exocrine gland, which now has the literal meaning of secretions outside. This is a key difference. Whereas the hormones are contained within the body and they're ductless, those glands, here we have ducts that secrete their products, specifically maybe sweat or oil, from the actual epithelial tissue to the surface of the epithelial tissue, outside of the body, exocrine. A couple of examples showing you the endocrine system. These are different glands. We have focused already in our class on the pancreas and its role in the secretion of insulin, for an example, when you have too high of blood sugar, or glucagon when you have too low of blood sugar. You can also notice these different glands here in the secretion of their products. And now exocrine glands. Again, exocrine secreting from epithelial tissue within to beyond the actual apical surface of your epithelial tissue outside. So that concludes our look at specific epithelial tissues. Again, you need to be well read and well studied on the specific epithelial tissues, their shapes, their layers, where you can find them, what they do, and how those functions relate to the structure of it itself. All right, well done. Go get them. We'll look forward to working with you in lab today.